So hello everyone, welcome to the NPTEL Immunology course for the June to November 2022 semester. I hope I am audible and my slides are visible. Uh, so, so I am Seemanti Aditya, a Ph.D. PMRF scholar in the Department of Biotechnology at Indian Institute of Technology, Madras. And I am a teaching assistant for the course Immunology, which is taken by Professor Agnew Ganguly and Professor Shudip Kumar Ghosh from Department of Biotechnology at IIT Kharagpur. So I will be taking live classes every Friday from 7 p.m. to 8 p.m. And here I will be discussing the concepts based on every week's content. And also, if you have any doubts regarding the concepts, we can feel free to ask me over here. So I hope I can start right now. It's almost 7:05. So yeah, so we'll begin. So the last class I had discussed about the overview of immunology, where we found out what were like the innate immune system and how the innate immune system and adaptive immune system are related, how the vaccination came into play, what was the history of immunology and all that. From now on, we'll go a little deeper and we will study in depth of all these courses according to the week's content. So this week we will be discussing about a little more details we will be seeing a little more detail in innate immune system and adaptive immune system, and along with that we'll also see some effector molecules like cytokines and chemokines and complements. We'll just go through the like overview of all of that. So let's begin. Uh, so so innate immune system, it is the first line of defense to all the pathogens. So why it is called the first line of defense? Because anytime any pathogen, if it enters our body, it is going to be first facing the innate immune system. And the innate immune system is present in almost all the individuals, and it is always there are always circulating macrophages and dendritic cells and neutrophils in the blood. It is not specific, so it is very non-specific type of immune system, and there is always a certain level of these cells that are circulating in order to protect ourselves from the surrounding microbes and the surrounding pathogens. So we are always exposed to so much of pathogens, right? So even after being exposed to so much of pathogens, we do not get infections very often. So this is because of the role that innate immune system plays. So any pathogen that tries to enter our body has to cross all these barriers of the innate immune system, and only if the innate immune system fails to remove those pathogens, then only the adaptive immune system comes to play. So the innate immune system has three main barriers to protect our body from the attack of pathogens. So these are anatomic barriers, physiological barriers, and phagocytic barriers. So anatomic barriers, as we have studied from a very long time, that we have skin, we have the mucous membrane on our, we have tears, etc. So any pathogen that tries to invade our body has to cross the barrier, has to cross this barrier, and skin provides a very good protection. So if there is any cut in the skin, if there is any scratch in the skin, uh, okay. So then the pathogens get a very easy access to the body, and infection can be caused readily. Otherwise, skin is a very important protective barrier. So the pathogens will first face these barriers, and their entry is restricted. If the pathogen somehow manages to cross the anatomic barriers, the second kind of barrier that will try to protect our body is the physiological barriers. So again, the pathogens will be opposed their entry into the body through the physiological barriers. That involves rise in body temperature, so we get fever, and there is acidity in the stomach. So this all criteria is essential for, it does not provide a proper environment for the pathogens to grow. So if even if it enters the body, because of rise in body temperature, because of this acidity in the stomach, it will not be able to survive. So as soon as it enters, it will be killed. So this is another barrier that will provide our body protection from the pathogens. And the third type of barrier, which is very important in case of innate immune system, in order to provide us immunity. So these are this barrier provides protection, and this barrier is a very important barrier to provide protection against the pathogen entry, and it is mediated by phagocytes, as the name suggests, phagocytic barrier. Now, phagocytes are any kind of cells that are able to engulf the pathogen and eat up the pathogen. So it will, as soon as the pathogen enters, non-specifically, it will chew the pathogen and it will kill it through phagocytosis. So this kind of barrier is mediated by phagocytes, and will kill pathogens by phagocytosis, and it will release inflammatory molecules. So inflammatory molecules are released in order to attract more and more phagocytes to that site of infection, so that whenever there are more cells, more number of pathogens can be killed quickly, and the immune system can function much more readily. So what are the key elements of the innate immune system? As we studied that there are physical barriers or anatomical barriers and chemical barriers. So this is the skin. Now, skin falls under the epithelial layers of skin and mucosal or glandular tissues. So, by the way, I am referring to Kubi's Immunology for all the slides or all the live classes that I will be taking. So, if you have any doubt, you can refer to QB. Yeah, so this is the skin, and skin provides the physical barrier to infection. Any breach in the skin or any tear in the skin will allow the pathogens an easy access to the inside of our body. So physical barriers and chemical barriers are there to protect our body from such entry by those pathogens. But anyhow, if the pathogens try to overcome those barriers and enter the cell, then what will happen is the pathogens either can be engulfed by macrophages, so it will cause the phagocytosis of this pathogen and the pathogen will be killed. So phagocytosis will lead to degradation, and the pathogens will be chopped up into small peptides. So those peptides can again be shown on the surface of macrophages and dendritic cells. Both of them act as a uh, phagocytic cell. So it will chop up the pathogen and it will cleave the peptides and express those peptides on their surface by MHC molecules. So these MHC molecules will then help in activating this adaptive immune responses. So dendritic cell, basically, as I discussed in the last class as well, so it will help in providing signals from the innate immune cells that there is a pathogen that has entered the body, and so we need some kind of specific immune responses to eliminate it. So then it will go and cause the activation of the adaptive immune responses. So adaptive immune responses are very specific to a particular pathogen, and that will result in the synthesis of antibodies from the B cells and T cell responses. So adaptive immune responses are from two cells, B cells and T cells. So B cell will help in the release of antibodies that will cause neutralization of specific neutralization of any pathogen or an antigen. And T cell responses help in the cytotoxicity. It will mediate cytotoxicity, or it will directly kill the pathogen or the affected cells. Mostly, the B cells kills the extracellular pathogens, and T cell will kill the intracellular pathogens. T cell mainly recognizes MHC class 1 kind of molecule, like virus infected cells, and those kind of stuff is mainly dealt with T cell. Not all kind of and mostly the extracellular parasites are dealt with this B cell responses or antibodies. So macrophages will go and chop the pathogens and present it to dendritic cell, and macrophages and dendritic cell will chop the pathogens and present it to the adaptive immune responses, and the specific responses are activated. Apart from that, pathogens can also bind to the cells, and these are normal cells. These are not any kind of antigen presenting cells or macrophages or any dendritic cells or phagocytes. These are normal cells, and whenever it binds to a normal cell, so it will get activated. So basically, all these pathogens will go inside the cell and start synthesizing its own compounds, its own RNA and protein, etc. So those proteins are not our own self proteins, right? So our body will be not synthesizing those proteins regularly if the pathogen is not present. So then those cells that have been attacked by this pathogen will get activated, and it will also show some kind of responses which can call the other phagocytic cells to its location and enable the killing of this infected cell. So it can do that by secreting mediators like cytokines and chemokines. So cytokines and chemokines are mediators. Why are they called mediators? Because this can, give a signal or give a notification to that to the immune cells that okay, there is a pathogen that has entered, and you immediately you need to come and clear that pathogen. So cytokines and chemokines basically are the signaling molecules, or they're also known as the mediators. And release of cytokines and chemokines leads to fever. These are the signs of inflammation. Cytokines and chemokines are related to inflammatory responses. So there are four kind of signs of inflammation that could be fever, pain, swelling, or redness, etc. So yeah. So after that, this inflammation, you are seeing that inflammation will lead to the recruitment and activation of protective cells and molecules. Also complement system. So complement is a very important protein. We will in the later on slides, or in the later on week lectures, we will be seeing how complement proteins are activated, and it provides important immune responses to the to eliminate the pathogen entering the body. And so this is also a part of the innate immune response. So complement system, macrophages, dendritic cells, cytokines, chemokines, etc. These are all the part of innate immune responses. Apart from that, also antimicrobial substances are synthesized by this activated cell, like peptides and interferons, that will directly lead to the degradation of this pathogen, and pathogen will be eliminated from the body. Also, if pathogens are infecting the cell, natural killer cells can kill those infected cells. So natural killer cells normally patrol the entire body, and they have the capacity to recognize infected cells through, I think, some kind of molecules or some changes on the surface of these infected cells. So it will go, and this is also a very non-specific kind of killing, where infected cells will be killed by natural killer cells or NK cells. So as we have studied that, so this all these responses are basically innate immune responses, the physical barriers, the chemical barriers, and all of this phagocytosis and degradation, the release of cytokines and chemokines, release of antimicrobial peptides, inflammation, the killing by the NK cells. These are all uh, the the activity of innate immune responses, and these are very non-specific kind of responses that we get. So innate immune responses is a very important role that is played is by macrophages, dendritic cells. So they carry out a process which is known as phagocytosis. So what is phagocytosis? Any kind of bacteria or any kind of pathogen that will enter our body. So this will be secreting, this will have on their surface some kind of molecules that are known as PAMs, or pathogen associated molecular patterns. And these PAMs are recognized by our immune cells like macrophages. They have on their surface what is known as PRRs, or pathogen recognition receptors. And this PRR and PAMs interaction will help in recognizing this antigen by the macrophages or dendritic cell, and it will result in the engulfment engulfment of this antigen inside the macrophage cells. So this is the bacteria that is getting engulfed. So we see that macrophages attacks the bacteria by recognizing the PAMs present on the bacterial surface through the PRRs that is present on their surface. Their surface means the macrophage's surface. So okay, so the full form of PAMP is pathogen associated molecular patterns. This is the full form of PAMPs. Okay. And PRR stands for pathogen recognition receptor. So yeah. So PAMs and PRR will interact, and PAMs and so that will lead to the engulfing of this pathogen, and so macrophages on their surface have toll-like receptors, toll-like receptors, TLR, and complement receptors that are known as the. Okay, somebody has raised a hand. Do you have any question? Anybody has any question till now? Okay, so we'll proceed if nobody has any question. So bacteria is basically endocytosed inside the endocytosed in the macrophages. So what happens is it will try to engulf the bacteria, and the bacteria will be present inside this macrophage in the form of a phagosome. So this phagosome is a phagosome where the bacteria is engulfed, and here after engulfing the bacteria and phagosome, we also have lysosome inside all the cells, and this lysosome has a lot of hydrolytic enzymes or acidic enzymes. So after engulfing inside the macrophages, once the bacteria is endocytosed in a phagosome, the lysosome will fuse with this phagosome. So what will happen is phagosome will be fused with lysosome, forming one big vesicle, and whatever bacteria is there inside this phagosome, due to the activity of all of these hydrolytic enzymes from lysosomes, it will get chopped off and it will be killed. So all the bacteria and all the pathogen that was engulfed will be killed. So fusion of phagosome and lysosome leads to the formation of phagolysosome, and this is an important component for killing the bacterial cell or the pathogenic cell through phagocytosis. So as lysosome contains many acidic hydrolytic enzymes, pathogens are killed by the phagolysosome formation, and this leads to the formation of activated macrophages. So all the macrophages which have already killed these pathogens are now activated macrophages, and this activated macrophages will present this processed antigen. So antigens are already processed, right? Here all the antigen that was engulfed earlier is processed, and they have small peptides now. So this will be presented on their surface by MHC molecules, and antigens will be presented to the adaptive immune cells by these MHC molecules. And we remember that we said we had discussed last class that T cell can only recognize antigens which are presented by an MHC molecule. It cannot recognize a whole antigen. So only if it is presented, if the antigen is processed and presented by an MHC molecule through an antigen presenting cell, it can recognize the antigen. So this will result. So all of this killing the cells, killing the bacterial cells will result in the formation of an activated macrophage, which will present the processed antigen to the adaptive immune cells and will release the cytokines as well. So this macrophage cells will present the antigen and also will release all the cytokines in the circulation. These cytokines will help in enhancing the immune response, because cytokines, remember, these are mediators and signaling molecules. So this will attract more and more immune cells to the site of infection and will help in clearing this infection. So there is a signal transfer that happens between the innate immune system and an adaptive immune system that we were discussing. So if innate immune system is not able to clear the pathogen on its own, then it will mediate this signal to the adaptive immune system. So adaptive immune system will come into the picture much later. But when it will come into the picture, it will try to have the immune response will be stronger than the innate immune response, because it is very specific to that particular pathogen. And also adaptive immune response, when it is activated, it can remember the pathogen for a longer period of your time, almost your whole lifetime. So a same pathogen, if it is entering the body next time, the adaptive immune response will quickly recognize it and quickly try to eliminate the pathogen through a variety of other mechanisms. Also, it is very important to remember that innate immune response and adaptive immune response, all they work in a very coordinated manner. None can function without the other, and there is no such like only one pathway that is occurring whenever a pathogen is entering. There are several pathways, several cells which are trying to protect our body from the attack. And we cannot say, so immune system is really a complex thing to understand. Immunology is not very like, okay, so there is not just one single thing that is happening. There is a lot of things that are simultaneously occurring. So important players. Now what is now let's come back to the topic. So important players which will allow the transfer of these signals are the antigen presenting cells and the chemical mediators. Now, antigen presenting cells are those cells which are directly coming in contact with the antigen or the pathogen, and it will interact with this antigen, and it will process those antigen into small peptides, and this is suppose the APC, and it will present the antigen to the adaptive immune response. This is the antigen presenting cell. So it is mediating the signal, like it is mediating the information to the adaptive immune response that there is a pathogen that has entered. Apart from that, the chemical mediators that play a role is chemokines, cytokines, and antimicrobial peptides or antimicrobials. So the professional APCs or the professional antigen presenting cells are macrophages, dendritic cells, and B cells. It is interesting to note that B cell, it is itself a part of the adaptive immune response, but it still plays a major role as an antigen presenting cell, because it will present the processed antigen to the T cells, and the T cell and B cell together will try to eliminate the pathogen in a more rigorous manner. So if you rank the immune system in a way, so T cell is the most dangerous kind of or the most highest level of immune response you can get from T cell. Then comes the B cell, and then comes all other innate immune cells. So macrophages, dendritic cells, and B cells are professional antigen presenting cells. They will present the antigen to the adaptive immune cells. So antigen presentation means that it is presenting this antigen to the adaptive immune cells, and it will result in antibody generation from B cells. Sorry. It will result in antibody generation from B cells and T cell mediated cytotoxicity. So T cell directly can interact with the antigen and kill the cells. B cells, on the other hand, will not be able to kill the cells, but it will release this antibody. This antibody will go and neutralize these antigens. Apart from that, we have chemokines, cytokines. So both of them are chemical mediators. And chemokines are mainly chemoattractants, or they help in chemotaxis. What do you mean by chemotaxis? So movement of the immune cells towards due to the release of chemokines. So chemotaxis is a general term, but in case of immunology, we are more concerned about the immune cells migration. So the movement of immune cells due to the release of chemokines to the site of infection. This is mediated by mostly chemokines. So these are chemoattractants. They will attract all the immune cells to that site, and it will promote cell migration. On the other hand, cytokines, this chemical is more prone to mediate cell-cell communication, because cytokines can be interleukins or interferons. So interleukins, interferons, tumor necrosis factor, etc. So cytokines are mostly associated with cell-cell communication. What does that mean is, instead of chemokines that are more concerned with attracting the cells towards site of infection, cytokines have variety of other roles. It will result in activation of certain cell. It will result in maturation of certain cell. It will result in the communication. So it is basically associated with the communication. And also, into some cases, cytokines and chemokines both are associated with migration of cells, but cytokines mostly is associated with this activation of cells, or signaling between the two cells. So, yes, somebody has raised a hand. Any question? Okay, somebody has asked that what will happen if an antigen will directly interact with the T cell and B cell without processing of the antigen. Okay, before that, another person has asked, innate immune system, one time explain. So innate immune system is any kind of non-specific immune responses that you are getting. So all the whenever a pathogen enters the body, and there is first line of defense, which is allowed by, which is conferred by the macrophages, dendritic cells, by engulfing those pathogen and clearing out these infections. Those are known as innate immune response. And this innate immune response helps in the synthesis of inflammatory molecules that I was discussing, like chemokines and cytokines, that will further help in increasing the innate immune cells to the site of infection, and eliminating the infection. And the second question is, what will happen if antigens directly interact with T cell and B cell without processing the antigen? So T cells and B cells, they need antigen process. Like B cells can directly interact with the antigen, but T cell will not be able to interact with the antigen without processing. It will need processed antigen presented on the surface of an antigen presenting cell through an MHC molecule. Then only T cell can recognize those antigen. But for a B cell can recognize a whole antigen and can produce antibodies after getting activated. And somebody has asked that, will you discuss practice questions related to assignments? I will discuss in the later on lectures once I complete this. So yeah, so initiation of the inflammatory response. So any kind of damage to the bacteria, sorry, any kind of damage to the body or will result in the resident senatal cells to release the chemoattractants. All the cells that are residual over that are resident over here, they will release chemoattractants and vasoactive factors, which will trigger a local release, a local increase of blood flow and capillary permeability. So whenever the skin is poked by a nail, or there is a wound that you have got, there is a scratch, there is a chance that bacteria will enter the system, and the cells which are very nearby to this site of infection will mediate this signal, and this will result in the release of chemoattractants. This could be chemokines or cytokines, and that will help in increasing the permeability of your endothelial cells that are present on this blood vessel. And the circulating cells, that are circulating immune cells that are always there in your blood, will then be directed towards the site of infection. And the neutrophils and phagocytes are the neutrophils basically are the first type of cells that are going to reach the site of infection. And all these neutrophils and phagocytes will then migrate to the site of infection through chemotaxis because of the release of the chemoattractants. Uh, and finally, phagocytes and antimicrobial substances will destroy the bacteria over here. So this will result in the initiation of an inflammatory response. So inflammatory response will lead to the increase of cytokines and chemokines secreted by those cells, and that will allow the infection to be easily cleared out. So what is inflammation? Whenever an immune cells are trying to get rid of the infection, they will release chemicals that result in migration of more immune cells to the site of infection. So thereby it will result in pain, swelling, rise in body temperature, redness, etc. This response is known as inflammatory response. Now, any kind of inflammatory response could be of two types, which could be local response and global response. So local response is when neutrophils, mast cells, and activated macrophages, these are the innate immune cells that are first going to reach the site of infection, and it is trying to clear out the pathogen by phagocytosis. And so they will release certain molecules known as leukotrienes, prostaglandins, chemokines, cytokines, etc. So all of these molecules, they are again chemokines, and these are chemical attractants, and mostly leukotrienes, prostaglandins, etc. They are the precursor of arachidonic acids, and they are associated with pain and swelling. So this will result in the local response. So wherever there is a site of infection, there is pain, swelling, and redness. But global response will result due to the local response, because because the neutrophils, mast cells, and activated macrophages are secreting leukotrienes, prostaglandins, chemokines, cytokines, etc. Global response will be activated. That will result in the rise in body temperature, swelling, not swelling, okay, rise in body temperature and feeling weak, etc. So this will be this is a systemic response that will cause that will cause an effect on the body level. Like you will feel tired, you will feel very lethargic after if you are being infected with any kind of pathogen. So how do the immune cells will migrate to the site of infection? This is an interesting concept to understand. That is extravasation. Before that, there is a question. Uh, Vasoactive factor, histamine, PGs. Vas Vasoactive factor, histamine, PGs, or any other. Yeah, so vasoactive factors. So what is vasoactive factors? They help in vasodilation, or like this we have we have this endothelial cells, right? So these endothelial cells, like those vasoactive compounds will help in increasing the dilation of your blood vessel, and also increase the permeability between these two cells. So those could be histamine. Those could be histamine is synthesized, secreted by mast cells whenever it is present in the surface, whenever it is coming in contact with bacteria. So mast cells will release histamine, and histamine will act on this endothelial cells to increase its vascular permeability, as well as it will help in dilating the blood vessels. So if you're dilating something, already you are stretching it, and so the permeability is going to increase. So that is the concept. And what is a vasoactive factor? So vasoactive factor is anything that is going to increase the dilation of your blood vessel. So, and opposite to that is vasoconstrictor. So vasoconstrictor will constrict the blood vessels. And the next question is difference between local and global responses. So local response is initiated in a local place, wherever you are infected, wherever you have the wound. So if you have a cut, or if you have an injection, so you will, if you will get a, if you have a cut, or if you have a like nail, a tear in your skin, so there you will see that there is swelling or there is redness. Even if you are having a mosquito bite, so there is swelling, redness, etc. So after the mosquito bite, some days later you will get fever. You will be weak, you will feel nauseous. So those are global responses, and that is mediated by the that is the reaction to all these leukotrienes and prostaglandins that have been synthesized locally to get rid of that infection. So global response is when you are experiencing any kind of fever, any kind of weakness. But local response is especially in the site of infection, wherever you are getting infected, wherever there is a pathogen entry. So the site of infection, basically. Yep. So we will proceed, and now we will discuss about how immune cells migrate to the site of infection. So immune cells, mostly neutrophils or macrophages, they can move from this endothelial cell, they can move from this blood vessel. So this is the lining of the blood vessel. Okay, lining of blood vessel. And as you have seen in the video also, sir had discussed, there are 4 steps by which this neutrophils basically gets activated. Then there is an adhesion step and transendothelial migration that will ultimately lead to the entry of this neutrophils or macrophages to the site of infection. Suppose there is a site of infection over here. So all these steps will basically allow the neutrophils, macrophages, and dendritic cell to squeeze through this endothelial cell and reach to the site of infection. So the first step, rolling, these are mediated by certain kind of cell adhesion molecules or CAMs. Cell adhesion molecules. And cell adhesion molecules, these are integrins, selectins, or ICAMs, as sir had discussed. So cell adhesion molecules are expressed on the surface of both the immune cells as well as the endothelial cells. Now, whenever there is so these are circulating cells, they will be circulating in the blood. Now, whenever there is a site of infection, there is a triggering of, there is a release of chemokines, cytokines, etc. So that will lead to this greater interaction between this cell adhesion molecules present on the surface of both these cells. So once there is an interaction, basically with neutrophils, what happens is there is a cell adhesion molecule called mucin, and there is a cell adhesion molecule called selectin. So mucin and selectin interaction will, selectin is present on the surface of endothelial cell, and mucin is present on the surface of the neutrophils. So mucin selectin interaction, due to due to the site of due to the release of inflammatory molecules, will result in the attachment, or the circulating molecules will get attached to this endothelial cell and will roll on the surface of endothelial cells. So because of the triggering the infection. So once this mucin and selectin gets uh, uh, gets to interact, there is a second kind of interaction that will take place between ICAM molecules that are expressed on the surface of endothelial cell. ICAM is immunoglobulin class immunoglobulin family of cell adhesion molecules. Sorry, family. Oops. Okay. ICAM immunoglobulin family of cell adhesion molecules. So ICAM will interact with another receptor, that is integrin, and this integrin and ICAM interaction, as well as the selectin and this is selectin and mucin. So this interaction will finally lead to the activation of these cells, and there is an adhesion to this particular endothelial cell. Finally, after this adhesion, there will be squeezing of the endothelial cells to the four different types of immune. Okay, somebody has asked what are four different ways of immune cells. Do not understand the question. Vasodilation occurs due to the release of all these vasodilating factors like histamine or leukotrienes, prostaglandins, etc. So yes, so this interaction will finally allow the neutrophils to open up this space between the two cells, and this once it opens up the space, this particular immune cell can squeeze through the space and reach the site of infection, and then carry on with its job. So here it is written that selectins, integrins, and immunoglobulin superfamily members, chemokines, etc., they are responsible for this entire process. Now, what are the effector mechanisms by which adaptive immune cells kill the cells? So, how T cells and B cells will kill the cells? So, both have different kind of approaches to kill the cell. B cells will produce antibodies, and antibodies help in neutralization of pathogen by binding to the pathogen. It has antigen antibody interaction, will help in neutralizing the pathogen. Next will be opsonization, that will allow phagocytosis of the pathogen. So what are opsonins? Again, I had discussed in last class. So opsonins are any substance that will enhance the phagocytosis. So molecules that will coat on the surface of bacteria, these are known as opsonins. These are known as opsonins, and they will help the phagocytic cell to recognize those recognize the pathogen and help in opsonization. Yeah, so antibodies will help in neutralization of pathogen. Opsonization, it will coat the surface of pathogen and allow the macrophages and dendritic cells to do their duty, and also help in complement activation. Now, complement activation, we are talking about complement proteins, that these are very important. So there will be lectures which will be wholly based on complement activation. It's a very important part of the innate immune system. So what are complement proteins in brief? Complement proteins are inactive proteins which are present in the circulation. Now, during any kind of infection, these complement proteins get cleaved. So suppose there is complement protein 1, complement protein 2, complement protein 3. So this gets cleaved into different subunits, C3A, C3B, etc. So this complement activation is due to the cleavage of this complement proteins, and this is associated with the formation of a membrane attack complex on the surface of bacteria. So whenever there is a bacteria, the complement proteins, after getting activated, C2A, C2B, after getting activated, they will form a complex. They are forming a complex of proteins, and this is a pore through which the cellular contents can be released outside. This is a pathogen, and it will cause the lysis of the cell. So this is a very important concept where MAC is formed, and that leads to complement activation. Then complement activation also results in opsonization. That is, complements can also bind to the surface of pathogen and allow the macrophages to do their job. And it can be of two types, that is antibody dependent and also antibody independent. Complement activation can be of two types. We'll discuss about it later. And how T cells mediate the immune response. So T cells are again of two types, cytotoxic T cells and helper T cells. So cytotoxic T cells will directly interact with the cell, cells like which are presenting the antigens, on their surface through the MHC molecule. So MHC molecule with the antigen recognized by the T cell receptor present on the surface of the cytotoxic cell. This cell will then release granenzymes and perforins. So perforins are associated with the formation of pore. So through this pore, granenzymes are released. And these granzymes will help in chopping off the cell and cause the cell lysis. Whereas helper T cells are not associated with cytotoxicity, rather it helps in differentiating into different several subtypes. There are several subtypes of helper T cells. There are Th1, Th2, okay, Th17, T regulatory cells, T follicular helper cells, etc. So all of these cells are associated with different functions, like activation of macrophages, then activation of the liver to secrete the complement proteins, and then T cell inhibitory responses, etc. So these are like helper T cells are not directly associated with cytotoxicity, rather it has different roles in different kind of situations. So then we'll answer some questions. So selectin and mucin, does it require any activation step? They are present always. Why not they interact in general? So yeah, so anyway whenever there is an infection, as I said that there is chemokines and cytokines, so already so there are circulating immune cells in your body. So once an immune cell will recognize any kind of pathogen, then it will send those signal molecules that will help this other molecules like neutrophils. First interaction with the pathogen is not always activated, because it is by chance. You are meeting a pathogen by chance. So similarly, this immune cells also will be circulating in their in the blood normally, and they will meet a pathogen. And as soon as they meet a pathogen, they recognize that this is not our body cell. So we have to inform this to the to our adaptive cells and other cytokines, etc. So they will release these factors, and these factors then will help that the selectin and mucin will interact with each other, and will increase their, so they can interact normally, right? But they are not interacting because they are always in the circulation. There is no such triggering factors like chemokines or cytokines that are secreted. So because of that secretion, this interaction is much more enhanced, and that will allow the passage of this human cells to the site of infection. The next question is, what is the thing that the mast cell do not secrete? What is the thing that the mast cell do not secrete? Okay, what kind of question? that is, I have no idea what kind of thing it does not secrete. What is T cell and cytotoxic T cells are same? So no, T cell and cytotoxic T cells are not same. As I said that T cells are two types, cytotoxic T cells and helper T cells. So T cells will include both the cytotoxic cells and helper T cells. So we have to be specific what cells you are trying to explain. So cytotoxic T cells and T helper cells both comes under the category of T cells. Next question is, how will B cell and T cell do the killing? That is what I am discussing. B cell is going to produce the antibodies, and T cells are going to kill the cells directly by secreting granzymes and perforins. So this is how T cells and B cells will do the killing. Next is difference between MHC class 1 and MHC class 2. So MHC class 1 molecules are expressed mainly on the surface of the MHC class 1 molecules are, identified by CD8 T cells. So these are cytotoxic T cells. And MHC class 2, these are recognized by CD4 T cells. Those are T helper cells. Now, what is MHC class 1 and class 2? So, MHC class 1 is expressed by all of the cells. MHC class 1 is expressed by all cells. Your own body cells, expressed by all cells. And MHC class 2 mostly expressed by antigen presenting cells. So, all these macrophages, dendritic cells, neutrophils, etc., they are going to express MHC class 2. We will discuss about major histocompatibility complex in details later. Major histocompatibility complex has like other roles other than major histocompatibility complex is very, again, an important part of the immune system. So this is histocompatible, means you are always you need to have this whenever a pathogen from other so other person's body will enter your body, so that is not your cell. Like even if it is from your same species, like humans, other pathogen is humans, other cells are entering your cell, like whenever there is a liver transplant or kidney transplant. So there is where this major histocompatibility complex play a major role, and it is we will study about this in details later on. Next question is, and what MHC1 will display on it, and what on MHC2. So both MHC1 and MHC2 is going to display on their surface processed antigens. MHC class 1 is mainly associated with viruses, because these cells are virus can attack any cells. So viruses will be attacking your lung cells or liver cells or whatever. So all these cells express on the surface MHC class 1 molecules. So whenever the virus attacks those cells, so this is processed by this is presented on the surface. They are not engulfing the cells. These are like normal cells of your body. But they will express this antigen, like some proteins of this antigen will be secreted, and then they will present it on their surface like with MHC class 1 molecule. But MHC class 2 is exclusively present most in the antigen presenting cells. So the antigens that are processed and engulfed by macrophages, dendritic cells, and neutrophils, those are going to be presented by MHC class 2. Now, next question is MHC necessary for T cells? MHC is necessary for T cells. T cells cannot recognize a whole antigen. It has to recognize an antigen which is processed by an antigen presenting cell and presented to the T cell using an MHC molecule. Now, MHC1 has one transmembrane, while MHC2 has two transmembrane and interacts with CD2, where MHC1 with CD8. So, yeah. Okay, in line, what is your thought? Okay. So, mast cells only synthesize and secrete histamine, proteases, prostaglandins, leukotrienes, heparin, variety of cytokines. Okay, so you are discussing among yourselves. Very good. How will T cytotoxic cell know that the antigen is being presented on MHC cell? Is there any toxin or chemotoxins released? How will cytotoxic cell know? So so we will discuss about like how T cell is getting matured. Okay, so let's start with T cell maturation. This is the last topic, and then we'll go for the questions. So T cells, they are generated in the bone marrow. Both T cells and B cells are generated in the bone marrow. Last class we had discussed that hematopoietic stem cells present in the bone marrow is going to generate all types of cells, including our immune cells. So the hematopoietic precursor will release the immature T cells. So immature T cells will then migrate to the thymus where it will get matured. This is the thymus, this is the bone marrow, and this is the blood. So immature T cell will then migrate to the thymus, and there it will get matured. So when a T cell is first generated, it does not have any kind of CD4 or CD8 on its surface. So these are marker molecules for T cells, and only if CD4 and CD8 is expressed on its surface, then only we can conclude that this cell is a T cell. So all the cells have its specific markers. So for T cell, it is CD4 and CD8. So initially, after being generated in the bone marrow and migrating to the thymus, it does not have any kind of expression molecules like CD4 or CD8. So these are known as double negative cells. DN stands for double negative. Okay. So double negative for both these protein molecules. So now, once it will undergo several steps of maturation, you see that there is DN1, DN2, DN3, DN4. So different steps, what will happen is the T cell receptor that needs to be expressed on the surface of the T cell. So T cell receptor has two chains, the alpha chain and the beta chain. So the T cell beta chain will go rearrangement and proliferation, and that will result in the commitment of the T cell towards being a T cell. So this was initially not committed to being a T cell. So once the T cell receptor's beta chain is proliferated and rearranged, so the T cell will get committed. And once the T cell is committed, there is several other steps of selection and proliferation. And again, a T cell receptor alpha chain getting rearranged, and all this DN1, DN2, DN3, DN4 helps in the expression of the T cell receptor in its proper form on the surface of the T cell. So once the T cell receptor is expressed, it will also express on its surface the CD4 and CD8. So it was double negative, now it became double positive. So it will express on its surface both the CD4 and CD8. So once the CD4 and CD8 is expressed, so these are known as double positive or DP, as shown here. So CD4 plus and CD8 plus, and then we have to select the cells. So there is two kind of selection, positive and negative selection. Okay, so there is two kinds of selection, positive and negative selection, that is based on the interaction of this. So a T cell is basically expressing both CD4 plus and CD8 plus, right? So now the interaction, how strong is the interaction of each of these molecules with MHC1 and MHC2 is what going to decide that whether this cell will be a TC cell or TH cell. TC cell has to have strong interaction with MHC1, and T helper cell will have strong interaction with MHC2. So MHC1 interacts with CD8 and MHC2 interacts with CD4, and this is TC cell, this is T helper cell. So wherever there is a strong interaction, this is affinity based selection. Wherever there is a strong interaction, the cell becomes committed towards that. The T cell becomes committed towards that particular subtype. So CD8 plus single positive cell is a CD8 plus cell, and CD4 plus single positive cell is a T helper cell. And other lineages which do not show any kind of interaction, they will die by apoptosis. And there are some other kind of lineages which have like T regulatory cell or T helper 17 cells, which do not go on like this positive or negative selection. These are some kind of exceptions. Negative selection undergoes when these kind of cells which have already been selected, if they interact with our self proteins. Okay, so any kind of cell proteins, so they will be rejected from the immune system, and they will be again pushed towards death, because now there is, if these cells are still remaining in your circulation, there is a probability of autoimmune disorders. So autoimmune disorders occur because your immune system cannot recognize your own cells. So your immune system is fighting against your own cells, and it is causing the death of those cells. So that is why this particular step is very important in order to prevent prevent such kind of autoimmune disorders. So now we will discuss some questions before I can discuss any other doubts that you have. I hope you enjoyed the presentation. Now we'll discuss some questions. So I'll basically share a poll. Please quickly look into your screen and answer those questions. These are really easy questions and based on what I discussed. Okay, great. So which are the first acting cells of the immune system? These are the neutrophils. As I discussed, neutrophils are the first cells that are going to go towards the site of infection and try to clear out the infection. Yes, Khushi, I have switched off my screen sharing. Now I am just sharing the poll. So you can see the poll and you can answer the questions. Now the next, okay, so I have some questions in the chat box. I'll quickly go through them. Okay, there are so many questions. This is what are the Yep. So Khushi has asked CD4 and CD8 both will be present on the same T cell. So during the T cell maturation, it is present on the same T cell. But once the T cell is matured, like the T cell that is circulating in your blood, that does not have both the receptors. Only one receptor is present, and that is going to designate whether the T cell is a cytotoxic T cell or whether it is a helper T cell. Okay, so either of the receptor is present during that maturation phase. That time both the receptors are initially expressed, and then one of the receptors are one of the receptors get eliminated. MHC.
Excuse me, ma'am.
Yes.
There's one question. The first cell taking its action into immunological response, isn't it macrophages? It's present in the tissue. It doesn't require any migration. So why neutrophil is being considered the first cells?
Because neutrophil, like macrophages are again, as you said, that is present in the tissue. So if there is any kind of like invasion which is present in a far away place, their neutrophils will be the first one to migrate because of its small size, I guess, that it can migrate very quickly and it can reach the site of infection.
Okay.
Yeah. Signaling can play an important role here. Yes, so signaling is very important in case of immune responses. MHC class 2 antigen present in cell, MHC class 1 present in cell antibody or antigen other cell mem. So MHC molecules are present in your cells, okay, or in your own cells, and antigens are expressed on the surface of MHC molecule, and those MHC molecules are recognized by the MHC molecule along with the antigen is recognized by the your immune cells or your antigen processed MHC molecules which are present on the surface of antigen presenting cell are recognized by the immune cells. And so They are present in two different types of cells. T helper cells have CD4, T cytotoxic cell or TG. Correct. Okay, and this will attach to a particular cell. Dear ma'am, how does memory cell memorize the characteristics of an antigen and recognize the next time? So whenever a cell is recognizing any kind of pathogen, B cell or T cell, irrespective of in this case, so B cell will recognize the pathogen and then it will differentiate into memory B cell and plasma B cell. So both of them are differentiating from the same cell that had earlier first time recognized the pathogen. So it is already having those receptors that have recognized the earlier pathogen, and once that cell divides into the two lineages, the memory T cell and this plasma B cells, memory B cells and plasma B cells, sorry. So the memory B cells have that information on its surface that okay, this pathogen looked like this, and I have receptors against those pathogen, and this is specific. Which cell will have only MHC on it? So our own body cells have MHC class 1. The cells which are antigen presenting or immune cells, they are expressing on their surface MHC class 2. Okay, so all the questions from the chat box is done, and now if well, whoever is remaining, I'll just post some other quick polls. So please answer those. I have already exceeded 6 minutes. I hope that is fine, and you don't have any other commitments. Yes, there are two memory cells, B cell and T cells. T cell and B cell will kill the pathogen, remaining B cell produces the antibodies. Yes, antibodies are produced by B cell, T cells are cytotoxic. How does memory cell memorize the characteristic antigen recognizer? Yes, Mahendra, I already answered that it is going to divide from the same cell that had earlier recognized the pathogen, so it will have the characteristics. Great. So how does a phagolysosome form? Obviously, the name suggests the fusion of phagosome and lysosome. Now, quickly, the next question. Functions of the memory B cells and T cells are same. The function is to eradicate the pathogen, but their mechanism of action is different. B cells is going to synthesize antibodies. T cell is going to directly attach to the pathogen and kill it. So I hope you guys understood what I wanted to convey. Right. So maximum of you have given the right answer. Migration of neutrophils to the site of infection across the high endothelial venule is known as extravasation. It has 4 steps that I discussed. I have posted another question. Quickly take a look. Somebody is presenting their screen. Okay, somebody, you have shared your screen. Rajkumar M. Right. So complement activation can lead to the lysis of pathogen by the formation of membrane attack complex, also known as MAC. So that is the correct answer. Most of you have given the correct answer. And the last question for the day. This is an easy question, right? T cell mediates what kind of immunity? So T cell is an adaptive immune response. It is a specific immune response. It is not innate immunity, and it is not non-specific. T cell is the highest kind of specificity that any immune cell can have. So what kind of immune response it is? Yes, it is cell-mediated response because it is directly interacting with the infected cell and causing cytotoxicity. So the cell will die, and therefore there is this cell-mediated immune response. So T cell is an adaptive immune response and a cell-mediated immune response. So that's all for today. So again, next class will be on next Friday from 7 p.m. to 8 p.m. Yes, yes, Shiva, I was talking about the polls. What are you? What is the questions? Okay, in the chat. What was the answer to the? What makes them? What was the answer to the second question? Uh, how does a phagolysosome form? So it forms by the fusion of phagosome and lysosome. So the name suggests phagolysosome. That is the answer. Somebody had asked. Okay, this cell. I think they have specific receptors. Okay, so I think you are done. Uh, Second time, third question share. Okay, I'll share the third question. So I cannot share the third question right now. The question was migration of neutrophils to the site of infection across the high endothelial venule is known as what? Migration of neutrophils to the site of infection across the high endothelial venule is known as the options were extravasation, vasoconstriction, endocytosis, and exocytosis. The answer is extravasation. So, yep, I think I am done for today. Endothelial venule is known as. Okay, so I hope you enjoyed the class. We'll again meet in the next Friday. Anybody has any other questions, we'll just discuss it maybe then. So thank you everyone.